Construct 1, Item 1. Scientists investigated cultured liver cells obtained from individuals with phenylketonuria, PKU. This item is sponsored by Masaka S.S. Big up to all the A-level students from Masaka. You are going to pass exams. Mutation of the PAH gene greatly reduced production of the phenylalanine hydroxylase enzyme. Excess phenylalanine accumulated inside the cells and interfered with several cellular processes. Some mitochondrial enzymes became less active, while amino acid imbalance reduced protein synthesis. Despite these changes, many liver cells remained alive. The affected cells responded by increasing anaerobic respiration, increasing glucose uptake, reducing protein synthesis, decreasing active transport. The investigation produced the following results. Pause the video, and analyze the parameters in the table. The investigation produced the following results. Pause the video, and analyze the parameters in the table. Task. Part A. Analyze the causes of the observed metabolic changes in the cultured liver cells, the relationship between ATP concentration and the observed cellular activities. Part B. Evaluate the effectiveness and limitations of the cellular responses in maintaining survival of the affected cells, justify the most suitable response, and recommend where necessary. Analysis. Causes of the observed metabolic changes in the cultured liver cells. Functional PAH enzyme decreased from 97% to 21% due to mutation of the PAH gene. Consequently, phenylalanine accumulated because it was not converted to tyrosine. The accumulated phenylalanine inhibited mitochondrial enzymes, reducing aerobic respiration and ATP production from 5.5 to 2.1 millimoles per kilogram. ATP concentration decreased from 5.5 to 2.1 millimoles per kilogram because inhibition of mitochondrial enzymes reduced aerobic respiration. Consequently, ATP-dependent active transport decreased from 95% to 46% because membrane transport proteins lacked sufficient energy. Lactic acid increased from 1.4 to 7.5 millimoles per kilogram because reduced aerobic respiration caused the cells to rely more on anaerobic respiration for ATP production. Anaerobic respiration produced lactic acid as a byproduct, resulting in its accumulation. Protein synthesis decreased from 100% to 52% because amino acid imbalance caused by excess phenylalanine interfered with normal protein formation. Consequently, fewer cellular proteins were produced, relationship between ATP concentration and the observed cellular activities. The decrease in ATP concentration was associated with reduced active transport. ATP supplies energy for membrane pumps, therefore reduced ATP lowered active transport from 95% to 46%. The decrease in ATP concentration was associated with increased anaerobic respiration. As aerobic ATP production declined, the cells increasingly generated ATP anaerobically, causing lactic acid to increase from 1.4 to 7.5 millimoles per kilogram. The decrease in ATP concentration was associated with reduced protein synthesis. Protein synthesis requires ATP for amino acid activation and peptide bond formation. Therefore, protein synthesis decreased from 100% to 52%. Despite reduced ATP concentration, many liver cells remained viable because alternative metabolic responses generated sufficient ATP to maintain essential cellular functions. Evaluation. Increasing anaerobic respiration. Increasing anaerobic respiration produces ATP independently of impaired mitochondria, enabling essential cellular processes to continue. However, it generates little ATP and causes lactic acid accumulation, which may inhibit enzyme activity. Justification. This response is effective for short-term survival because it maintains a minimum ATP supply when aerobic respiration is reduced. Increasing glucose uptake. Increasing glucose uptake supplies more glucose for glycolysis, increasing ATP production through anaerobic respiration. However, ATP production remains low because mitochondrial aerobic respiration is still impaired. Justification. This response is suitable because increasing substrate availability sustains ATP production needed for survival despite reduced respiratory efficiency. Reducing protein synthesis. Reducing protein synthesis lowers ATP demand, conserving energy for essential survival processes. However, prolonged reduction limits enzyme production, tissue repair, and normal cellular maintenance. Judgment. This response is suitable because conserving ATP allows limited energy to be redirected to processes essential for immediate cell survival. Decreasing active transport. Decreasing active transport reduces ATP consumption by limiting the activity of ATP-driven membrane pumps. However, prolonged reduction disrupts ion balance, nutrient uptake, and waste removal. Justification. This response is suitable because conserving ATP helps sustain essential metabolic activities when energy availability is limited. Overall recommendation: increasing glucose uptake is the most suitable cellular response because it directly increases substrate availability for ATP production, enabling continued energy generation to support the other survival responses. However, reducing phenylalanine accumulation remains necessary for long-term restoration of normal cellular metabolism.